Hi, so for our final paper and presentation, we decided to look into one's genetic predisposition to alcoholism and alcohol sensitivity. This project was done by me, Kayla, Cayanan, Chris Collins, and Jordaine Tran.
Hi all, so today I'm going to be giving an introduction to our topic on the role that genetics play in alcoholism and alcohol sensitivity. So in order to understand the research that we'll show you later on, we first need to define what alcoholism is and its impact. So alcohol use disorder is the impaired ability to stop or control alcohol use despite adverse social, occupational, or health consequences. And alcohol use disorder is also colloquially known as alcoholism. And so we found out that approximately 95,000 deaths per year are attributed to alcohol. And this makes it the third leading preventable cause of death. And then, just for some background, the first is tobacco, and then the second is poor diet and physical inactivity. Then also, alcoholism, the economic impact is alcohol misuse costs the US 249 billion. And the criteria for this economic impact are lost productivity, healthcare costs, and criminal justice costs to the courts and prison. And that is not to mention the fact that you can't put a cost on a human life. So now I'm going to be talking about what contributes to alcoholism and alcohol sensitivity. So, alcoholism, much like many of the disorders and diseases that we've covered this semester, is not a single gene disorder. It's polygenic. This means that there's not one specific gene in the genome that is responsible for someone becoming an alcoholic. Instead, there are actually many biochemical pathways that are involved, some of which include serotonin transporter genes, the dopaminergic reward pathways, which we'll discuss later, is kind of implicated in the learning pathway. So alcoholism can kind of the reward that you get from alcohol can be learned and lead you to become addicted. So that's how the dopaminergic reward pathway is involved. Also, the GABA receptor pathways are involved, and there can also be deficiencies in alcohol dehydrogenase, which is an enzyme responsible for the breakdown of alcohol in our system. Also, there's a large and complex number of environmental factors involved, much like many of the disorders that we've covered this semester. So then here on the right is the GABRB3, the gamma aminobutyric acid receptor subunit beta 3, which, as we'll see later in some of the research, is implicated in alcoholism. During our research about alcoholism, we identified three themes. And the first is alcohol sensitivity, second is alcohol dependence, and the third is about alcoholism. So now I'm going to go into the first theme of alcohol sensitivity. So the first line of research that we found was involving the serotonin transporter gene 5-HTT-LPR, and researchers found this to play a significant role in sensitivity to alcohol. So those who were homozygous for the L allele had lower intoxication scores. So if you have this homozygosity for this allele, alcohol did not affect you as much, which could lead to the possibility of a development of alcoholism. They also found that early life stress correlates with a decreased alcohol sensitivity. And this was a study done on rhesus macaques monkeys. And early life stress in that study was defined as not growing up with their mother. So the monkeys were antisocial, and that was the early life stress category. The next research that we found was about the alcohol dehydrogenase. And alcohol dehydrogenase is an enzyme involved in the breakdown of alcohol. And Hasiba et al. In 2019 found that chronic alcoholics have a higher alcohol elimination rate. And so basically, that's just how fast your body works through alcohol. And this is in part due to the enhanced alcohol dehydrogenase 1 activity. Heath et al. In 1999 also found that an increase in alcohol sensitivity correlated with a decreased risk of alcoholism. And this should make sense, right? If you're more sensitive to alcohol, you don't have to consume as much, right, to experience the intoxicating effects. So the increased sensitivity was correlated with the decreased risk of developing the problem of alcoholism.
The second theme we're going to be discussing is alcohol dependence. A study performed by Grant et al. Analyzed genetic comparisons of alcohol-dependent symptoms while also analyzing the corresponding amount of consumption in individuals that are not alcohol-dependent. Alcohol factor scores from the twin subjects were shown to have been moderately heritable with a genetic correlation in accordance with alcohol-dependent symptomology. Results indicated that there is a moderate heritability of alcohol-dependent symptoms, increased risk of alcohol abuse in individuals with corresponding genetic correlations for reported alcohol-dependent symptoms, and increased risk of heritability in individuals with alcohol-dependent symptoms and increased consumption. Another study identified 282 SNPs from 61 genes were identified to be involved in serotonin, dopamine, and GABA pathways, specifically in Taiwanese Han men. So they found significant genetic associations with alcohol dependence in familial subjects, as seen here. Increased genetic risk exists in the Taiwanese Han population in genes involved in metabolizing alcohol. The third theme is alcoholism. Researchers examined the genetic effects of specific polymorphisms of alcohol dehydrogenase and aldehyde dehydrogenase. These are both enzymes that are able to break down either alcohol or aldehyde. Subjects who had ADH2, ADH3, and ALDH2 were found to be less frequent in Taiwanese Han men who had alcohol-dependent symptoms. Researchers noted that there's still a complex result of both environmental factors and genetic predisposition. The GABRB3 or GABA system genes, CA repeats, were found to have a significant association with alcoholism. It was found that more severe alcoholics had a larger number of CA repeats when compared to control and less severe alcoholics. There was an increased risk of alcoholism severity when gene variants of GABRB3 and DRD2, which is a dopamine system, were found in individuals.
Now I'm going to be going into the conclusions based off the studies that we read and future directions for future studies. Based on the studies we read, evidence seems to suggest that there is a genetic influence on the development of and one's predisposition to alcoholism and alcohol sensitivity. This is due to biological, genetic, and environmental factors. When we read the studies, we found that some of these factors included serotonin transporter genes, early life stressors, aldehyde dehydrogenase genotypes, and other various gene variants that contribute to one's likelihood of having decreased sensitivity to alcohol, which could lead to an increased risk of developing alcoholism and alcohol dependence. Additionally, there seems to be no significant impact of having a family history of alcoholism, according to the studies that we read. In terms of future studies, there are two directions that we want to address and take. The first direction is utilizing gene therapy. There hasn't been much study or research on using gene therapy for combating alcoholism. However, we think that this could be an effective way for anyone who's battling alcoholism or has an alcohol dependence to target their disease. And so how we would go about doing this is doing a study that has 3 groups, and these 3 groups would be a group of people that would have no therapy, a group of people that would undergo cognitive behavioral therapy, and a group of people that would undergo gene therapy. In gene therapy, how this works is a short hairpin RNA will target liver cells in order to inhibit the release of an enzyme that aids in changing ethanol into acetate. This metabolization of ethanol into acetate is a key step in the process of breaking down alcohol. And so a vector that is carrying that RNA would be injected into the cell for those in the gene therapy group. And so we think that this would be an important type of study to conduct to see the difference in the therapies and see which one is more effective, and if gene therapy does seem to have an impact on alcoholism, and see if it could improve one's dependence in some sort of way. The second direction that we would take is to recruit a more diverse sample group. And a lot of the studies that we read, they focused on specific groups, and which made it a little bit harder to generalize the study that they conducted to the entire population. And so because of this, we would like to recruit in a wider area or population. A lot of people who undergo or who are affected by alcoholism are within the age range of 18 to 24 years old. And so we think it'd be important to recruit at a university, especially a university that seems to have a diverse demographic, and then also within neighborhoods with different socioeconomic backgrounds, and not just in white rich neighborhoods. By having a more diverse sample group, it will allow our results to be more easily generalized to the wider population, which will make the research much more effective in understanding alcoholism and how it impacts people from different backgrounds. And so these are the future studies that we would like to conduct and the future directions we'd like to take in order to better understand this relationship with genetics and alcoholism.